Thank you very much for that very nice introduction. And then I will just start with my presentation. Um, so CD8 T cells are critical components of the adaptive immune response, and they have cytotoxic properties, which means they can eliminate virus-infected or cancer cells. And there's two main types of CD8 T cell responses. There's acute responses, which are formed to low levels of antigen, and CD8 T cells can then massively proliferate and also acquire effector function. And there is CD8 T cells that encounter high levels of antigen. And then in these scenarios, which is, for instance, chronic infections or cancer, over time, CD8 T cells acquire a lot of inhibitory receptors, which dampens their proliferation and also overall effector function, including the production of cytokines, interferon gamma, and TNF. And overall, so there's these two main types of infection, and the subsets of CD8 T cells in response to each are also quite different. In response to acute infections, when the cells are encountering lower amounts of antigens, they form effector cells, which have high killing capacity but are short-lived, and they form memory precursor cells, which can then give rise to memory T cells, which persist after the infection has been cleared. And if the same pathogen reinfects the host organism, these cells can mount a faster immune response, even more robust than the primary one. Whereas in contrast, in response to chronic infection, there's also a formation of short-lived effector cells that express some level of inhibitory receptors, such as TIM3, but they have to form precursors of exhausted T cells, which have self-renewal capacity and also can differentiate to more to exhausted cells that have some level of effector capacity. So these cells are overall considered to be dampened in their effector capacity, but they still exert some control over virus or tumors. And the capacity of the precursors of exhausted cells, so the TPEX, to self-renew is critical to sustain the response over a prolonged period of time. And so when we compare memory and TPEX cells, we realize that they do have some similarity in the sense of they both have capacity to self-renew and maintain themselves, and also give rise to more differentiated populations. And this maintenance of this stemness program means that they don't necessarily acquire effector properties, so there's a suppression of effector programs. But memories are formed in the absence of antigen. They respond essentially to homeostatic proliferation from cytokines such as IL-15 and IL-7, so they are considered to be in a quiescent state. Whereas the precursors of exhausted cells, they are formed to high loads of antigen. They have to be constantly proliferating under the T cell response stimulation because they are constantly encountering antigen stimulation, and they give rise to constrained and exhausted progeny. So they have similar properties, similar functions to say, but they are selected by very distinct environments. And considering that some infections have an acute stage of development followed by a chronic, our question on a very essential level was whether there is potential in the acute infection to generate a chronic response. And we also asked if such potential was to be present, were it restricted to a specific subset of cells. And one of the tools that we have in general to study CD8 T cell responses is the lymphocytic choreomeningitis virus infection, which causes systemic infection in mice, which control is dependent on CD8 T cells. And there's two strains of LCMV that can be exploited. One is an acute LCMV, Armstrong, which leads to a low antigen infection that can be rapidly cleared, thus leading to the formation of memory cells, and also a chronic LCMV challenge, that infection that leads to high levels of antigen and a chronic challenge. So in this setting, the cells will become exhausted. And to better target the CD8 T cell response, we can also transfer P14 T cells, which have a TCR specific to the GP33 LCM to GP33 epitope of the LCMV, so they respond to the infection. And what we did to answer our initial question was to do single cell RNA sequencing on P14 cells isolated from mice exposed to acute infection on the early stage of infection, so day 8, and also on the memory stage of the infection, so day 21, and compare them to P14s exposed to a chronic LCMV infection, also on day 21 of the infection. And here you see that PD1, which is an inhibitory receptor, and TOX, which is a very important gene for exhaustion, they are expressed in the exhausted populations, but not in the cells that come from the acute infection. And TCF7 and the IL-7 receptor genes can be identified can be used to identify memory precursors, the memory cells on the later stages of the acute infection, and also the TPEG cells when responding to the chronic infection. And what we noticed was actually that in response to the acute infection, there are some cells that overlap with the TPEG cells from the chronic infection on day 21. And so we could essentially distinguish two subsets of memory precursors, and we compared their gene expression, identifying that the more classical memory precursors that do not overlap with the TPEG cells express CXCR6, whereas the ones that overlap with the TPEG cells express high levels of ID3. And so we then validated whether we could also find these cells based on flow cytometry. And so we gated within the TCF7 positive cells, TCF1 positive cells, which are memory precursors, distinguishing them from more effector cells that express KLRG1. And within these cells, we could identify ID3 expressing populations and CXCR6 expressing cells. So both at the MP2 and the MP1s in the lymph nodes and in the spleen. And we also could identify ID3 expressing cells on the memory stage of infection within the CDC12 expressing central memory cells, which are the memory cells that are considered to recirculate in the lymphoid organs. And also considering that our ID3 cells overlapped with the TPEG cells in the RNA sequencing, we also measured the PD1 and expression of TOX in these cells and compare them to the TPEG cells isolated from chronic infection on day 8. So both subsets, of course, express lower levels of these markers compared to the TPEG cells on the chronic setting, but the ID3 cells consistently showed higher expression of PD1 and TOX, suggesting that these precursors may be formed in response to encounter of higher amounts of antigen within the acute infection. And after identifying this subset, our next question became, what is the relationship between these two subsets in relation to each other in response to the acute infection? And for this, we transfer the ID3 positive CXCR6 negative memory precursors and the ID3 negative CXCR6 positive memory precursors to isolated from acute infection to infection matched mice also on day 8, and we did a readout on the memory stage of the infection. And what we saw was that CXCR6 positive cells actually expanded more than the ID3 positive cells at this stage. But when we looked within the memory precursors that were formed, the memory population based on the IL-7 receptor expression on these two populations of memory precursors, the ID3 positive cells could generate ID3 positive memory cells, whereas the CXCR6 precursors did not generate ID3 positive memory cells. So we concluded that the ID3 positive memory cells form the ID3 positive memory T cell pool in response to the acute infection. But that let us work the other way around. And so finally, we evaluated how these ID3 positive memory cells that come from the ID3 positive memory precursors compared to the ID3 positive, ID3 negative cells, also and the double negative cells, how do they respond to acute and chronic challenges? And so what we saw is that both subsets of the CD62L cells, so the ID3 positive and the ID3 negative ones, respond very comparatively to an acute infection. They both expand very comparatively. Whereas when we infect the mice with a chronic infection, after they have received these subsets of cells, we see that the ID3 positive cells expand more than the ID3 negative cells or the double negative cells, which are more effector-like. So that is sort of expected. And when we characterize these cells on a phenotypic level, we saw that only the ID3 positive cells could give rise to ID3 positive, TCF1 positive TPEC cells. So in response to the chronic infection. So only this initial memory population that we transferred to naive mice and infected could form the actual TPEC cells that we know to be critical for the maintenance of the response in chronic challenges, whereas these other populations seems to form primarily effector cells that decline over time more rapidly, whereas these one have the capacity to self-renew and generate more progeny. And so what we concluded on the resolution level was that we have an ID3 positive memory precursor that can give rise to ID3 positive CD62L memory. And on an acute level, these cells seems to fare comparatively. So they seem to both be quite adapted to generate responses to an acute infection. But when there is a chronic challenge, only the CD62L ID3 positive cells can give rise to TPEC cells, the actual PD1 expressing TPECs, and sustain the immune response thereby. And so the question to us then became if the ID3 expression itself is required for the maintenance of the response to acute or chronic infections. And for these, we transferred ID3 deficient P14 cells compared to control P14 cells to mice and then infected them. And what we saw was that the ID3 deficient cells show decreased expansion in both acute and chronic LCMV, but the response in acute was still far more comparable in terms of proportional decline compared to the chronic. And therefore, this matches with our observations that the ID3 cells are rather dispensable in the acute infection, but truly required to sustain the response to chronic challenges and add the information that ID3 expression is required. We saw that in the ID3 deficient cells, there was a decline in the TPEX population as early as day 5 post-infection that was also sustained over time. So on day 21, populations were still declined. And we performed RNA sequencing on this population of TPEX cells, comparing the ID3 deficient ones to the control TPEX. And we saw that genes related to cell stemness, such as CD62L and MIB, were actually enriched in ID3 deficient TPEX, whereas control TPEX seem to have more robust gene programs related to proliferation and metabolic function. And what we observed that indeed this control TPEX had decreased expression of CD62L, which are considered a more undifferentiated stem-like population of TPEX. So they are earlier in the differentiation of the population, and reduced expression of these KIT-positive TPEX that you can see here when you look over time in the chronic infection, in the control mice, so the black ones, the frequency of KIT cells peaks on day 12 of the infection, which is also when the cells are cycling more. And And therefore, the reduced expression of KIT also associates to some extent with a reduced proliferation of TPEC cells. So our overall conclusion was that these TPEC cells do seem to be formed, and they form the CD62L populations, but then these CD62L populations are somehow defective when ID3 is not present, and they can't really expand properly, and that's why the response is overall so reduced in ID3 deficient P14 cells. And to really show this, because we had a defect quite early on day 5 post-infection, and so we wanted to pinpoint that the defect is on the TPEX population, and also suggesting that they can't really proliferate. And so for this, we transferred the control and the ID3 deficient P14 to separate groups of naive mice. We infected them, and then we sorted the population separately, and we co-transfer them into the same mice. But so at this stage, they are already TPEX cells, and we transfer them to infection matched mice and perform the readout on day 12 of the infection. And so we saw that even when the TPEX are co-transferred, they are still massively decreased in the ID3 deficient cells, leading to reduced populations of TPEX and TEX. And when we evaluated the expression of P67, which is a marker related to cellular proliferation, we see that at this peak of the infection, the control TPEX in the same mice have high expression of P67, whereas that is massively absent in the ID3 deficient TPEX cells. And that's not the case for the TEX population. So it's really we believe it's really a defect on the capacity of the TPEX cells to expand that leads to an overall decline of the CD8 T cell response to chronic challenges. And we also try to see if that's also the case in response to cancer. So we used a melanoma model that expresses GP33, so the virus antigen. And then in mice that had been inoculated with this tumor line, we transferred control or ID3 knockout P14 and evaluated the tumor growth. And the tumor escaped on a control level to the ID3 deficient cells, whereas the control P14 showed increased capacity to control the tumor and also increased survival of the mice, suggesting that ID3 is required in both the chronic infection and also in overall response to cancer. So in both exhausted settings as the cells encounter high levels of antigen. And given how adaptable our ID3 populations seem to be in mounting a response to chronic challenges, we are also interested in if we could improve ID3 expression in vitro. And so we performed the cytokine screening, whereas we identify that treatment with IL-36 beta and IL-18 leads to increased ID3 expression in T cells generated in vitro compared to IL-2, but also to other cytokines that we have on the manuscript as well. And we also demonstrated that whereas, for instance, expression of expansion under IL-2 treatment is independent of the ID3 expression, expansion with IL-36 beta or IL-18 is dependent on ID3 expression. So IL-36 beta and IL-18 boost ID3 expression and also select for cells that express ID3 in their expansion. And so we try to see if this treatment of cells could be a strategy to improve cancer control. So the idea was that we inoculated mice with B16, so melanoma lines, but expressing ovalbumin as an antigen, and we transferred activated OT1 cells that are specific to ovalbumin that had been pre-treated with different cytokines on day 7 post-tumor inoculation. And on day 13, post the cell transfer, we saw that in the tumor draining lymph nodes, which are a very important site for the anti-tumor response, that we had increased frequencies and numbers of OT1 cells. So they had increased TPEX essentially, and that led to an improved tumor control and survival of the mice over time compared to mice that had received no cells or had received cells pre-treated with IL-2. And so we overall demonstrate that we have this lineage of cells that express ID3 and can respond to a high to responses that can respond to high loads of antigen, both by being preformed in the acute setting or being immediately differentiated into TPEX cells, and that the proliferation of these cells in response to to high levels of antigenic chronic challenge is ID3 dependent, and that these cells can be the differentiation of these cells can be favored by treatment with IL-18 or IL-36 beta, whereas other cytokines may be favoring the differentiation of more classic memory precursors. And with all this, I would like to acknowledge everyone that has worked with me, particularly Daniel, who was mine and Mania's supervisor, and who is the main leader of this story and who developed the main questions that we answer, and also Axel Callis and Rick Johnson, who led several different aspects of this story and were involved in a lot of discussions. Amanya and Dane are both first authors with me on the paper, and this was really a collaborative study. And I want to also thank everyone who works with me. And finally, thank you all for listening. I will be happy to